Click for IB Biology. This is our second video on the organelles of eukaryotic cells. In this one we'll be looking at the mitochondria, the chloroplast and the Golgi apparatus. There is a PDF down in the description which allows you to make your own notes at the same time as you watch this. It's blank, so you can just annotate that. There's a 10-question quiz with answers so that you can check your understanding of the information that I'm relaying here. Now let's recall that the purpose of these videos is to allow us to identify these structures in the electron micrographs produced from electron microscopy. Details on each of these structures, these organelles, are to be found under the cell specialization videos. So this purpose of this is to be able to recognize mitochondria in an electron micrograph, and here's an excellent image of an electron micrograph of a mitochondria given here on the left hand side here. So the features that we should be looking out for can be referred to in the diagram here, and the most obvious first one is the presence of an outer and an inner membrane. So we have two membranes. We can clearly see this at this item here. Now the inner membrane is folded inwards to increase surface area, of course, and we can see these folds in regions like so. Of course we can see them all over the image, but here are some cristae here. So these folds are called the cristae. Now the functioning of these different regions of the mitochondria is covered in great depth in the cell specialization videos, but also in the parts of the syllabus associated with respiration, aerobic respiration. And that's a reminder that the function of the mitochondria is to synthesize ATP. It makes ATP, and it does so in an aerobic manner, which is to say that it uses oxygen. That oxygen is taken in and after circulation is transported to your cells, where it will be taken up on the cristae membrane to actually absorb hydrogen to form water. So that gives you a clue of some of the respiratory biology which is to follow. But recognizing a mitochondria, we look for the folded inner membrane. The center is called the matrix. The matrix is where a piece of chemistry takes place called the Krebs cycle, named after the person who first published the information. One or two other things about mitochondria is that they have their own little loop of DNA, much like a prokaryotic cell, and they also have their own ribosomes, which are actually called mitoribosomes, and they also are very prokaryotic-like, and they're contained within the mitochondria, and that's where they stay. The length of a mitochondria can be up to 10 micrometers. The diameter, if we were to see this in cross-section, then there would be half a micrometer to 1 micrometer. It depends. You get different sizes and numbers of mitochondria in a cell. So back to the image here, we can see the double membrane. We can hypothesize that the matrix is here. We know that the cristae are folded. That's where oxygen goes. We need to understand the process in more depth to appreciate that. Double membrane, and we know that this length here is somewhere perhaps less than 10 micrometers. And just for reference, these structures here are in fact endoplasmic reticulum with ribosomes stuck around the outside, also here as well, just for size comparison. The mitochondria is where we synthesize ATP. That's the correct way to explain the function of a mitochondria. Let's compare that to a chloroplast, obviously a feature of plant cells, and we carry out the process of photosynthesis. So we have a diagram here that we can compare to the micrograph to help us in our identification. And once more, notice the double membrane around the organelle. Not quite so clear in the image of the chloroplast in the micrograph, but there is a double membrane there. And then these dark line structures are the thylakoid membranes. With the general grayed space here is the stroma. Now the thylakoid is where we find chlorophyll, and the purpose of these membranes and its chlorophyll is to absorb light energy. And that light energy is then transformed into chemical energy, which actually takes the form of ATP and NADPH. That sounds complicated, but later on when you study photosynthesis, this will be made very clear. So the light energy is turned into the chemical energy of ATP on the thylakoid membranes here. This energy is then used in the stroma, where we find an isolated, the container here of chemistry is the Calvin cycle. And in the Calvin cycle, this is where, amongst other things, we get the formation of sugars. And in this case, oxygen is a waste product of that process from the splitting of water. Sizes approximately the same as mitochondria, can be up to 10 micrometers, usually about 0.5 to 1 micrometer in diameter across this dimension here. And then having made the sugars are stored often here. These are starch grains, and one can see roughly in various parts of the stroma small circular structures. Perhaps you'll be able to see them on your video channel, which are actually lipid oil droplets, because this is a plant as well. So this is the chloroplast, only seen in plant cells, of course, double membrane. It also has its own DNA. It also has its own ribosomes, and they remarkably resemble bacterial cells. More about that later. The site of photosynthesis. And then let's take our final organelle for this video is the Golgi apparatus. This curious structure named after the person who hypothesized its existence. Golgi was a histologist who hypothesized that there would be within a cell a structure responsible for taking proteins and modifying them by adding things like sugars to them to form structures like glycoproteins. He hypothesized that this structure would exist, and finally it was observed. Golgi never actually saw this structure himself. The stacks of membranes are about 1 to 3 micrometers in depth, kind of going across here like this. We can see these membranes stacked on top of each other. That's a phrase which is used. They're often known as Golgi stacks, and it's referring to all of these membranes on top of each other. So in there, proteins are being modified, having sugars added, having lipids added to make lipoproteins, before they are actually exported from the cell. So these are not for internal use, but for external use. So more details about that in another video. So the mitochondria, chloroplast, and Golgi apparatus. Our final video on eukaryotic cell structures, the link is now showing. There's the link to the channel should you wish to subscribe, and of course to the playlist with all the details for this particular unit of the course.